Okay, well, good morning or good afternoon. My name is Manan Sharma. I'm a research microbiologist at the USDA Agricultural Research Service. I work in the Environmental, Microbial, and Food Safety Laboratory in Beltsville, Maryland. I'd just like to thank the organizers and the conveners of this session and this meeting for inviting me to speak today to talk a little bit about persistence of pathogens in soil and in irrigation water. Following up on what Dr. Ingram led off with, and some of the regulations around it. Today we want to just talk about how some of these things get into the pre-harvest produce environment, like is what we like to call it. And essentially what we're talking about is we know that we're using inputs that contain bacterial, parasitic, and viral pathogens. And what we want to do is utilize the nutrients and the natural resources to grow produce and to recycle some of these nutrients so they don't wind up in the environment and causing deleterious effects. We want to use those nutrients, but we want to leave the pathogens behind. And we know that manure and irrigation water can contain many different bacterial, viral, and parasitic pathogens. Today, we're just going to focus on the bacterial ones, but that's just my expertise, and the parasitic ones can also be important. When we're talking about bacteria on produce in pre-harvest environments, we're talking about bacteria that are not growing in a mammalian gut. They're not in warm-blooded environments. They don't have access to nutrients. They're subject to a lot of different stresses like sunlight, microbial competition in soil, which isn't the same in the gut. There's a lot of ultraviolet radiation from the sunlight. It's a lot more dry than they're used to in the soils without rain. The dynamic temperatures between days and nights, or cooler and hotter seasons, are not what they're used to. So this is a lot of physiological stress for them to survive in. And we have the saying that all produce safety is local because it's dependent on the soil and the type of crops you grow and the weather and what type of manure you might use. But a more technical way to say that is all pre-harvest produce safety is affected by the interaction of spatial, temporal, agricultural, and weather factors, because it's really hard to dismiss the agricultural effects and sort of the location and the weather effects. We know we have a lot of different sources of surface water contamination. This is a slide I got from the EPA, and we're probably going to focus more on the cropland and the animal feedlots in this picture here. But we know that rural homes, city streets, suburban developments, these are all can be sources of various pollutants into the waterway. And when we have flooding events like we had last year in Iowa and in the Midwest, and that can cause a lot of contamination as well, spreading these pathogens from one field to another, or even into the waterways. So there's a lot of different ways to get things into water and potentially onto food that we eat. And the thing about tracking bacterial pathogens in pre-harvest environments is like these pathogens, when there's an outbreak, they don't leave like they don't leave a confession note, okay? They don't say was here in irrigation water after being in animal manure, and then we were transferred to crops that were consumed raw, and that's what caused this outbreak of infections. You're doing a lot of investigative microbiology here, and you're trying to backtrack to figure out, well, it was here, where do we think it was before, and how did it get on this lettuce that was contaminated, or these cantaloupes that was contaminated? So there's really not ever a clear picture. There's not ever a really nice flow chart that shows it went from point A to B to C to D to E to F. It's a lot of finding and then putting the pieces together. And we know in previous outbreaks that RV septic tanks have been a presumed source of bacterial pathogens to contaminate lettuce. We know that concentrated animal feeding operations have also been presumed sources of pathogens. And in both cases, it was presumed that these sources contaminated irrigation water that then subsequently contaminated growing crops. And for example, with a CAFO, what you could have is two different mechanisms of contamination. You could have manure that actually runs off into irrigation water and contaminates the water and gets transferred to the crop, or you could have a direct transfer mechanism where there's manure dust that's generated and then transferred to the crop. So manure dust generated here and then transferred to the growing crop here through sort of a more direct transfer. For some work done in our lab, we think of the water as a vector. The water is just transporting this pathogen from A to B. But some work done in our lab and some other work done at the University of Vermont with Deb Neer's group basically showed that these pathogens have the ability to grow in simulated manure runoff. And this was runoff that we created, that we simulated using a very common biological soil amendment of animal origin that Dr. Ingram talked about. These are heat-treated poultry pellets. This is a common a common fertilizer that organic growers use. We basically made some simulated runoff with this, inoculated it with a pathogen Salmonella Newport. And if you look at our chart here, the blue lines is our runoff that's made with the heat-treated poultry pellets. The orange line is just soil with none of those poultry pellets added. And you can see that the Salmonella can actually grow to a much higher level in the amended runoff, in the runoff that contains the poultry pellets, than in just the soil alone. So this is one way that once it gets into the water, if the conditions are right and the temperature is right, you can have growth of these pathogens. Some other work done in our lab by Ruth Oney, who now works for the FDA, but she evaluated the survival of Salmonella on spinach plants if they were introduced basically in dust particles or if they were introduced in water particles. And this red line is the Salmonella survival in the dust on the spinach leaf. The black lines are the Salmonella survival in the water on the spinach leaves. And what you can see pretty clearly from this graph is that the decline of the Salmonella in the dust is much, much slower and much smaller than it is for the Salmonella in the water. So what this indicates is that if you have that dust that's transferred and has a pathogen in it, those pathogens can survive for a much longer duration than if it's actually in water. So the matrix that it's transferred on also matters. One of the projects that we've spent a lot of time in our lab working on is called Conserve. This is a USDA NIFA-funded project led by the University of Maryland with very many collaborators from the University of Maryland College Park, the University of Maryland Eastern Shore, the University of Delaware, the University of Arizona. And this is basically a 360-degree look at what's in our water. I can tell you more, but if you go to the website www.conservewaterforfood.org, you can learn a lot more about what we do here. And this is just to show you that what's in our water varies by region and by pathogen. Excuse me. Varies by region and by pathogen. So, sorry, went ahead a couple of slides. So varies by region and by pathogen. I apologize for that. And basically, if we're looking for Shiga toxigenic E. coli, which is E. coli OM57H7, what we have is a really low prevalence across the country from 0.2 to 14%. But if you look at Salmonella and Listeria monocytogenes, you see much higher prevalences based on the location, the geographic region, on the types of water sources that you're sampling in, ponds, rivers, lakes, maybe it's treated, recycled wastewater. These are all things that affect the prevalence of these pathogens. And just to take again the Shiga toxigenic E. coli, you can look and see what the geographical difference makes. This is a really large study done in California by my ARS colleagues, Michael Cooley and Lisa Gorski. They had almost 1,400 samples in a watershed in California. They found about 11% of their samples contained Shiga toxigenic E. coli. When we did a similar study here region in Maryland, we only found about a 2.4% prevalence of Shiga toxigenic E. coli here. So again, the types of water, the geographic location, what animal, livestock inputs, what the climate is, all of these things can affect the prevalence of these pathogens in the water. We also conduct a large survey here as part of the Conserve project, where we looked at between 10 and 35 sampling events over 2 years at different sites ranging from rivers and creeks to ponds and reclaimed surface water. And the main takeaway from this study was that basically we found much higher prevalences of Salmonella and Listeria monocytogenes in rivers than we found in ponds. And this is kind of surprising to me because when you looked at these ponds looked really not well kept. They had a lot of moss, a lot of algae. There was many things flowing into it. You would think that just from the visual appearance of the pond, oh, it has to be contaminated with something, but it turns out they're much lower prevalence than, say, like the rivers that we looked at, maybe because there's less agricultural inputs, maybe the ponds are rain-fed, maybe they're there to hold groundwater. I should mention that these are agricultural ponds, and some other authors have found the same thing as well in Virginia and Georgia. So in general, ponds had less pathogens than the rivers, at least the ones we sampled. In another large study that we did, a 4-year study where we looked at survival durations of E. coli in different types of manure-amended soils, what we did was we looked at 3 different research farm sites in the Mid-Atlantic, 2 in Maryland and 1 in Pennsylvania, and this is the map of that region, with different soil types. And basically we tracked 2 different types of E. coli here. We would apply different types of manure to different types of soils, inoculate it with E. coli, and basically look at how long this E. coli could survive in these soils, just to see what matters. Does the manure type matter? If it's poultry litter or horse manure or dairy manure liquids or dairy manure solids, does it matter if it's tilled or surface applied? Does it matter if it's managed organically? Does it matter what year and what site and what season we do this experiment at? We're trying to evaluate all of these things to help support the FSMA rulemaking process about the intervals that you need to have between the application of raw manure or untreated BSAAO and the harvest of crops that are going to be consumed raw. So basically, this was a really big study, 12 separate field trials over 4 years. We got about almost 6,700 data points, 324 survival profiles. These are what those survival profiles look like after some really nice modeling done by our statistician, Brian Vineyard. And the takeaways from the study were that in general, poultry litter amended soils supported longer survival durations of E. coli. We were looking for the survival time, not the actual levels. So we're looking at the survival durations of E. coli. And poultry litter supported that longer survival duration compared to horse manure or unamended soils. But really what we call the spatial temporal effects, the site, the year, and the season where the study was done were extremely influential in determining that survival duration. And our FDA colleagues in the Decisions and Risk Sciences branch took our same data and looked at it a little bit differently. They were looking at the actual levels of E. coli in our studies. And what their take-home messages were in this paper were that the amendment type, so poultry litter again, predicted longer survival durations, but also longer survival levels. E. coli survived at higher levels for longer periods of time than the other manure sources, dairy and horse manure or unamended soils. But the other thing that was interesting here is that the days of rain since the previous sampling day was also pretty influential. And if you're familiar with a random forest output, this is a variable importance plot here showing you the different types of factors that went into their model. So in this case, days of rain is the number of days that rainfall actually occurred. It's not actually the total amount of so these are interesting things, and that's really good federal data sharing here. We feel really good to collaborate with the FDA with their ability to do this type of analysis, and they originally supported this work through an interagency agreement, which we really appreciated and allowed us to do such a long-term study. Some other work that we have not published yet, but we just completed and submitted was with, again, a collaboration between the University of Delaware and the FDA and USDA ARS. And this is work led by Dr. Pushpinder Litt and Dr. Callie Neal's lab at the University of Delaware, basically looking very similar to our other study where we look at soils amended with poultry litter, composted poultry litter, heat-treated poultry litter pellets, or a chemical fertilizer. We take all of these, apply them to the soil, add our E. coli, similar to what we did in the previous study, and then we plant cucumber plants in that soil. And over 120 days, we take soil samples, and then when the cucumbers are ready to harvest, we take cucumber samples and look for levels of the E. coli to see what happens. And in this case, what we're concerned with is the amount of transfer that we get from the cucumbers, from the soil, which had this E. coli, to the cucumbers. And you can see that we've done this in 2 years, in 2018 and 2019. These are different biological soil amendments of animal origin that we're evaluating here, but that's less important than what these black lines are right here. And this is a box and whiskers plot. This black line is essentially the median value, 50% above, 50% below. And you can see here that in 2018, our median values of all their different treatments are much higher than they are in 2019. So again, this goes back to the role of year and season on the survival of pathogens in the pre-harvest environment. And this differs a lot by year, and you can see that we're getting some decent transfer here of the E. coli that we know that we put in the soil, and it is transferring to these cucumbers. Still a pretty low level, 2 to 3 logs CFU per cucumber. But in 2019, we just have a couple of outliers that get that high. The median values are basically just at 0, right above 0. So what accounts for this difference in year? Well, if we look at the total amount of rainfall and the rainfall patterns, both of these things I think are important. So we think the total rainfall during season may affect transfer. And if you look at here in 2018, we got about double the amount of rainfall than we did in 2019. But it may not just be that rainfall amount. It may actually be the pattern that this rainfall is organized in. So you can see here in 2018, you can see that we don't get a lot of rainfall. This is rainfall measured over 10 days, a cumulative amount of rainfall every 10 days. And you can see there's not a lot of rainfall in the first 30 days. But then what happens is we go up here between days 31 and 40, and we get really large rainfall days here between days 31 and 40, all the way up to 61 and 70. And almost the opposite happens in 2019, where you get more rainfall at the beginning, but then those rainfall amounts tend to fall through the study. And this, again, may affect the survival dynamics of the pathogens in soils. And what we'd like to say is that it's a combination of all of these things. It's the rainfall, it's the soil, it's the location, and it's also the type of biological soil amendment that you're putting into the soil that's affecting the survival of these things. So it's a very dynamic environment. And just to conclude, this is basically what we say. The spatial temporal effects can affect how long pathogens survive in soil. They can be transferred in water and then eventually transferred to a growing crop. And these are dependent on spatial temporal factors, but also specific agricultural effects, like the soil type, like the manure type, like the crop being grown, whether it's grown on the floor, whether it's high or low to the ground. And really what we're looking for are agriculturally predictive variables that can predict the survival of E. coli, Salmonella, Listeria in these matrices. Is there something, a nutrient level, the rainfall amount, a specific chemical indicator, or a non-microbiological indicator that a grower could actually use to say, hey, my crop might be more at risk of contamination this season compared to others, or when I see this level go high, I need to pay more attention to the microbial contamination issues that may be occurring. And we're hoping that some of these large studies that we're doing can inform on those types of variables. But we really have to do these large studies first, go back and retrospectively apply them, and then do smaller studies evaluating those specific factors to see if this actually works out. So with that, I'd like to thank all the funding sources that have been instrumental in doing all of doing all this work. Our ARS base funded project, the Conserve project funded through NEFA, the interagency agreements funded by the FDA, and also our partner institutions, the University of Maryland Eastern Shore, the University of Delaware, and we have many collaborators at many different institutions across the country, North Dakota State, University of Maryland Eastern Shore, University of Delaware, University of Maryland College Park, and also our great staff here at USDA ARS. And with that, I'll conclude my talk. I just want to thank you all for your attention, and I'm happy to answer questions in the roundtable.